So today, I'm going to teach you guys how to do DNA extraction right at home. Sounds fun, right? Okay, so for this experiment, you're going to need some shampoo, some isopropanol, which you'll have to put into the freezer for about 20 minutes. You'll need about 250 milliliters of water. You'll need a small pasture pipette, which is fairly common found at home, a graduated cylinder, which you could use the pasture pipette if you can't find a graduated cylinder, and a mashed up banana. You could use either half of a large one or a full small one, or if you don't want to use banana, you can use about 3 to 4 strawberries. Remember to add about half a cup of water and smash it in a Ziploc bag. If you don't want to use a Ziploc bag, then you can put it in a blender, but this is in case you don't have a blender. And you'll need some salt. You'll need about 1 or 2 pinches of salt. Okay, so let's get started. So this is the shampoo that I'll be using. It's Tresemme. You could use any kind. The isopropanol, or otherwise known as rubbing alcohol. This is just the isopropanol, just pre-frozen. Then you have the graduated cylinder, the pasture pipette, a beaker with water, about 250 milliliters of water should do it, some extra beakers, which you could use anything that has measurements on it, like a measuring cup if you really need to. This is my mashed up banana in about half a cup of water, and some salt grains. Okay, so let's get this experiment started. So first, what I'll need is about 25 milliliters of water. So I'll take one of my beakers and find the 25 milliliter mark and measure it. Or you could do this using the graduated cylinder. It works the same way. Then I have to add in 1 to 2 pinches of salt. So I'll do that now. This is to stop the next ingredient, which is the shampoo, from foaming. So I'll also need 1 teaspoon of shampoo. And stir it until all the salt and shampoo has dissolved in it, dissolved in the water. Make sure that your shampoo does not foam up. And avoid making many bubbles. I know bubbles are fun, but try not to make bubbles. Depending on the kind of shampoo that you used, mine was a white shampoo, so it looks like it's foamed up, but it's really not. But once it's dissolved, it should look semi-clear to foggy. Okay, so once you have this dissolved, the salt and the shampoo, then you take up your mashed up banana and you take about 5 to 6 teaspoons and add it to this solution. So I'm going to just add my banana. When you mash it up, make sure that the banana and the water mix very nicely. And once again, instead of using a bag, you can use a blender, which would probably do a better job. I'll just add 6 to try and get enough DNA to show you guys what it actually looks like. once you have the banana in here, you have to stir for about 5 minutes. So, once you've stirred it for about 5 minutes, this is what it should look like. You should have some bubbling at the top. That's just your shampoo. Okay, so you have a filter paper, okay? Now you have to fold it. So you make one fold like this, match up the ends to make a semicircle, like this. Then you fold again from corner to corner to make a cone. Now what you have to do is take the first flap and open it to open up your cone. And you can fold it the other way just to make sure it stays like this. Now you need a funnel, and then you put your filter paper in here. And what I normally do to keep the filter paper in place is I usually take a bit of water. Well, first I need a beaker. I usually take a bit of water and I wet it. That allows for the filter paper to stay on the funnel. So I'm going to use this see-through glass. Well, so once you've wetted your filter paper and gotten it stuck to the funnel, now what you do is you take this mixture that you've stirred for a good 5 minutes and you put it through the funnel and you let it drain. So I'm just going to put it right through. I can't get all of it in right now, so I have to wait for some of it to filter through. But if you look closely, you can see that some of it is dripping through the funnel, little drops. That's going to be my filtrate that I'm going to use later on in the experiment. Okay, so once you have a few milliliters of your filtrate, which is this cloudy stuff that's in here, that's shaking, you take about a few milliliters using your small pipette, and you add it. So I have about 2-ish milliliters in here. That should be enough. And you add it to your isopropanol. Look at all that cool stuff. But once you get all of it in here, let it sit for a few minutes. This stuff that you see at the bottom, that's just your filtrate right now. After about a minute, you'll have a bit of stuff precipitating down here. That's not going to be your DNA. Your DNA is actually going to be going into your alcohol. So if you look closely, you can see bits and pieces of bubbles that look like bubbles that are surfacing at the top. That is your DNA coming up. And you can see some fibers and stuff near the bottom. That'll later separate from your DNA. So I'm just going to find the cap and put it onto the side to kind of let it sit for a few minutes. Okay guys, so we left the DNA for close to an hour, and this is what it came out to be. You'll probably see that at the bottom, I don't know if you guys can see it, there's some yucky stuff that's at the side of the test tube. That's not DNA. If you look closer at the bottom, there's like a sticky sort of syrup thing at the bottom. That's also not the DNA. Now, if you look at the top, you see this whole web thing up here? That is your DNA. Now, the thing is that the experiment itself says to leave it for a few minutes. If you were to leave it for just a little bit of time, this wouldn't be this thick. The fact that we left it for a whole hour is what actually got so much DNA out. Now, another way that you can get out more DNA is if you actually add more of your filtrate that you originally had. I didn't actually keep that much filtrate, but if you add all of it in there, you should be getting more DNA, but that's a sufficient amount. And that wraps it up for the DNA extraction lab. Subscribe to me so you guys can see some cool videos doing science and experiments and maybe learning something new. Thanks!